Vanakkam. Welcome to this course on classics in neuroscience. In the last few videos, we were looking at cortical columns and cortical processing, the neural circuitry associated with these. In this video, we will look at the auditory cortex and a possible auditory map, and we will also discuss the pattern theory of olfaction or smell. We discussed the number of studies that was performed on the visual system and some studies that were performed on the somatosensory system. But what about the auditory system? It is also a sensory system. What about the auditory system? It was shown, for example, by one Bexy that there was frequency selectivity along the cochlear basilar membrane, that is laid down by the travelling wave. Also, this line of work was then pursued by Archie Tunturi, who mapped specific frequencies in the auditory cortex. That is, there is this frequency selectivity for specific neurons. However, these studies were early studies and that were considered somewhat controversial because all these studies are performed under anesthesia, and it was later realized that this mapping is highly sensitive to the actions of anesthetics. So, there was a need to come up with newer methods, better methods that are still being pursued, that are still subjects of active research. What about olfactory stimuli? Something to keep in mind regarding the olfactory stimuli is that presentation of these stimuli cannot be as precise as presenting, say, a light stimulus or say a somatosensory stimulus, because that smell, how much smell in what quantity is going to come from what quantity of the chemical is somewhat difficult to control. So, this is a challenge in this line of work, something to keep in mind. This line of work was pursued by Edgar Adrian, who started studying the brain mechanisms involved in vertebrate olfaction. He performed multi-unit recordings from the olfactory bulb. These are not intracellular recordings, these are also not single unit recordings, and he found that different odors, those odors are elicited by different chemicals, acetone, xylol, pyridine, eucalyptus, produced different patterns of firing. The same set of neurons from which you record, they produce different patterns of firing. This idea came to be called as the pattern theory of odor representation. So, this essentially suggested that there existed a clear relationship between olfactory epithelium and the olfactory bulb. So, each chemical or each odor essentially elicited a signature pattern within the olfactory bulb. So, each of them have a specific signature or a specific set of activation. That set of neurons have a specific set of activation for that particular odor. So, this is called as the pattern theory of odor representation. But the question is, what caused this, this kind of patterning? What is the reason? What is the science behind this? What caused this kind of patterning? It was suggested by Mosel that this kind of patterning may be due to different gradients of absorption of the different chemicals that came to be called as the chromatographic theory of spatial patterning of odor responses. Mosel essentially suggested that this patterning may be due to different gradients of absorption of the odors. Finally, this line of work was pursued by Frank Sharp using a newer method, 2-deoxyglucose method, in which he showed that this was due to selective glomerular activation. These works happened much after the 1950s, that are the focus of our course. These are all the references that we have used in this set of videos, that is on neural circuits on cortical columns and cortical processing. Please do check them out. In this video, we looked at the auditory cortex and a possible auditory map, and we also discussed the pattern theory of olfaction. With this, we come to the end of this video. Thank you for your attention.